Hello students. In the previous lecture, I have discussed about some key terms of genetics like chromatin, chromosome, DNA and gene. Now let me take you to a more detailed terminology of chromosomes. Well, now you know how 2-meter long chromatin get condensed and packed up inside the cell nucleus of just 6 microns. You also know chromosomes are made up of DNA and histone proteins, and classical X-shaped chromosome structure is more visible at the metaphase stage of cell division. But just before duplication or replication, a chromosome looks like a single linear strand, just like this, a single linear strand with only two arms. One is this arm, one is this arm. We can call them as unduplicated chromosome. So this is an unduplicated chromosome. Okay. Now look here. This is a duplicated chromosome. This is a characteristic X-shaped chromosome, which can be called as duplicated chromosome, which contain two identical strands. One strand is here and one strand is here, and these two strands are joined by a centromere. This is a centromere. So this is a duplicated chromosome which contain two identical strands joined by a centromere. We can call these strands as sister chromatids. So this is a non-duplicated chromosome or chromosome just before duplication or replication, and this is a duplicated chromosome which contain two identical strands which are joined by a region which is known as centromere, and we can call those strands as sister chromatids. We human beings are organisms with diploid cells. That means, with exception to sex cells or gametes, our somatic cells contain two copies of chromosome. One we inherited from our father. We can consider this green one as the chromosome we inherited from our father, and one we inherited from our mother. This blue one is inherited from our mother. So we have such 22 pairs in our cell, all our somatic cells, which are identical in size, shape, and staining properties or staining pattern. We can call these identical chromosomes as homologous chromosome. Okay, so homologous chromosomes are basically identical in their size, shape, and staining pattern, and they are inherited from both of our parents, from mother and our father. So they are paternal chromosomes and maternal chromosome. So now it is clear that chromosome can be seen in pairs in diploid cells, both in unduplicated and duplicated condition. In duplicated condition, sister chromatids will be clearly seen. Now look at this chromosome. Based on the position of centromere, this is centromere, we have discussed it, the length of the arms differ. See, this is a short arm and this is a long arm. That is based on the position of the centromere. We can call the long arm as Q arm and short arm as P arm. So short arm is also known by the name P arm and long arm is also known by the name Q arm. It is important to see the position of centromere to identify a chromosome. chromosome can be distinguished on the basis of size and relative location of centromeres. Now look at this image. You can see 4 types of chromosome here. So in the first type of chromosome, you can see the centromere exactly at the center of the chromosome. So we can call that kind of chromosomes as metacentric chromosomes. But look at the second image. The centromere position shifted from the center, and you can clearly see short arm and also long arm. I hope you remember the names. Long arm is also known as Q arm and short arm is also known as P arm. Okay, P arm is also known by a term petite. Okay, and this is the condition which is known as submetacentric. And on the third type, that is acrocentric. See, the centromere position is moving towards the end. It doesn't reach the extreme end, but almost it reached. So here you can see the P arm or the short arm and a very long Q arm, right? And the fourth type is the telocentric chromosome. Telocentric chromosome is very special in the position of the centromere. See, the position of the centromere is the extreme tip of the chromosome, and this telocentric chromosome is not seen in human beings. We have metacentric, submetacentric, and acrocentric chromosome. So I hope now the classification of chromosome based on the position of centromere is clear to you. One is metacentric chromosome, other is submetacentric chromosome, third one is acrocentric chromosome, and fourth one is an extreme condition which is known as telocentric chromosome. And there is a point to remember. The extreme tip of the chromatids, see this position, is known as telomeres. Telomeres. And this telomeres is a specialized region related to cellular aging. So we will discuss about the telomere in some other video lectures. It's very important region which is acting as a protective cap of chromosome. Well, let me help you to identify a chromosome location. See this image. Here we have an X chromosome. Okay, it is written there. This is X chromosome with almost 900 to 1600 genes. And we have a gene here, PCDH11X. So we have to locate this PCDH11X. Actually, the location is given here. I'm going to interpret this location. It is written as X, that is X chromosome, and Q is there, that means it is located in the long arm of X chromosome. And what is this 21? Actually, the 21 is the region of Q arm or the long arm. This is the 21st region. Let me mark it with a marker. And after a period, you can say a period here. After the period, there is another digit is there, and that is 3. 3 is the band. So we can interpret this location as in the long arm of X chromosome in the 21st region and third band, you could see a gene which is known as PCDH11X. Okay, so it's very simple, but you must know what is that Q, what is that region, and what is that digit that signify after period. Okay, so XQ21.3 means X is the chromosome, Q is the arm, 21 is the region, and 3 is the band. So that is over. Now listen, the entire chromosome cannot stain uniformly. There will be some difference. I'll show you what's the difference. Like some region stains darker and some region stains lighter. What does that signify? Actually, in this image you can see the darkly stained region and also a lightly stained region. Darkly stained region is more condensed and have transcriptionally inactive or silenced genes. What does that mean? Because due to strong packing, genes get silenced on that particular darkly stained region. Here the darkly stained region shown is actually lightly stained. This orange color region I am talking about the darkly stained. So here genes are silent because it is packed very closely. For a better understanding, I will show you another image. This is the tightly packed region. So their genes will be transcriptionally inactive or silent, and that region will stain better or darkly stain, and that region is known as heterochromatin. So darkly stained region which is more condensed and have transcriptionally inactive or silenced genes due to strong packing is known as heterochromatin. So the region centromere and also the region telomeres are heterochromatin regions. Other regions in between, I will take another marker. Other regions in between this region is a lightly stained region and is less condensed and have transcriptionally active genes due to loose packing. This is a loose packing condition, and we can call that region as euchromatin. So euchromatin is a lightly stained region, that means less condensed region, and have active genes. So they are transcriptionally active region of the chromosome. So now you able to distinguish between the terms heterochromatin and euchromatin. Always remember, heterochromatin is a region which is transcriptionally inactive. That means genes are silent there. Euchromatin is a region which is transcriptionally active. That means genes are very active there. So the packaging difference also visible here, and there is another term here, acetyl group. When acetyl group is attached to the histone proteins, the packing get loosened, and we can call that kind of mechanism as epigenetics. We will discuss about epigenetics in some other video. Okay, so moving finally to an image which is very popular, and you are going to see this image in the upcoming videos very frequently, and we can call this image or pictorial representation as karyotype. So let us see what is a karyotype. In this image we can see an individual's collection of chromosomes in a diploid cell. This is the collection of chromosome in a diploid cell. Okay, there are 22 pairs. I'll take another marker. There are altogether 22 pairs of autosomes or You can call this as autosomes. Autosomes or non-sex chromosomes. Non-sex chromosomes. They are referred to numerically. See, 1 to 22 is there, based on a decreasing order of their size. That means the chromosome 1 is larger than chromosome 2, and chromosome 2 is larger than chromosome 3, and likewise. And chromosome 22 will be very small when compared to that of chromosome 1. So if somebody is asking you which is the largest chromosome in your body, you can very easily say that is chromosome 1, and it must contain so many genes also, as size is big, too big. Now what remains is 2 of our chromosome, which is sex chromosomes. A pair of sex chromosome, or we can call that as allosomes. Here it is XY. What does that mean? This is a male karyotype. How could I understand this is a male karyotype? That is a speciality of sex chromosome. If the sex chromosome is in the pattern of XY, that will be male, and if the sex chromosome is in the pattern of XX, that will be female. Wait, we are at another topic now, that is sex determination, and we will discuss that topic in an upcoming video. So stay tuned.